Hey, this is Mrs. Fano again. This is second part of my talk about the vaccine. So the first part, we talked about our body defense system. So there are three of them. One is the innate immunity, which is non-specific. Our body, everybody has it to defend our body's enemy in general. And they don't recognize particular enemy, just anybody that's not your like foreign will be defended. We'll try to get rid of. And then the second immune system, that's the second level, is the humoral immunity response, which is body fluid response. Those are the B cells. They will capture the enemy, analyze them, and then produce antibody to defend them, more like a foot soldiers, and they have guns, they have knives, and whatever to defend their enemies with antibodies. And then the third one is cell-mediated immune system response. That's the third defense system. That's when your enemies actually invaded your body, occupy your cells. So they will try to get rid of those cells that's infected. That's more invasive because you have to kill your own cells as well. Next, I'm going to talk about how the vaccine works. So first of all, we need to understand antigen and antibody. What is antigen? What is antibody? Antigen and antibody are like a key and a lock. The relationship between them is like a key and a lock. So antigen is more like the key, and then the antibody is a lock. So there's B cells, they will be on the cell membrane, but there's the floating in their body, those type of antibody, those will also travel around. So those will also bind to the antigen. So only particular antigen, not the other shape, only one type. One antibody only binds to one type of antigen. There are actually, for antibodies, there are three different functions. The first function is called neutralization. What does neutralization mean? For virus, they want to invade your cell. They want to get on your cell membrane and then get inside of your cell and use your cell's machinery to make their own protein or their own genetic information. So that's their first act. So that's what they want to do. So neutralization means the antibody will bind to the surface of the virus, especially that particular part that has a signal, tells it how to bind to the cell membrane, the cell surface. So if you can block that, then the virus cannot really bind to the cells, and then they are useless. They cannot invade your cell. So that's very important. That's for virus. And then for some toxin, the poisonous protein, if those antibody can bind to them, so they cannot actually be poisonous anymore. So that's called the neutralization function, what they do. And then there's another one called, let me see, opsonization. I hope I pronounce it correctly. It's a way when the antibody binds to the invaders. Here looks like a bacteria. It could be virus as well. So it will bind on it, and then it signals the macrophage to gobble it up, to eat it, to take it inside of the macrophage. And then or neutrophil, another cell that can do the same thing. So this is just an example for macrophage. And then that will sometimes, most likely, the macrophage will release some enzyme to just destroy it. So that's another way of destroying enemy. And then the third one is mainly for the bacteria. When the antibody binds on the surface of the bacteria actually make holes in them. When it makes hole, the bacteria will fall apart and then it will just die. So that's the three different functions. But for the coronavirus, is the first two. That's the first two. That's what the antibody does. If you have coronavirus, neutralizing antibody will bind to the virus surface, the spike protein basically, the most important, and then to prevent it from entering your cell. And then the other way is the antibody can bind to this also the spike protein, and then the macrophage will gobble it up and destroy it as well. So there's two ways to destroy the virus. Here's another picture about the macrophage. The macrophage's job is not just garbage disposal. It also has other functions. First of all, it takes in the bacteria and then it will gobble it and then digest it, use it almost like a food. So lysosome is the one that release enzymes and then to digest it. But in the meantime, it actually very smart, kind of writing a report, formula called MHC molecule. So that's one we've seen before for the B cell as well. So, and also release the antigen, which means the further the next time the bacteria, whatever on the like a signature on the bacteria will get recognized and released to the cell surface. So this is different way of telling your immune system to how to get rid of the bacteria. So this is important for the cell-mediated immune response. Remember, the T cells will recognize this surface, the MHC, this report. You'll take in the report, say, hey, what do? I need to make more maybe for the bacteria, especially important for have the killer cells because they're huge. The antibody may not really block them to do bad things. So they need the killer cells to actually kill the bacteria. So they are cells, they are not virus. So that's very important for that.